Hi, I'm Matthew Addison. I work for the apple, pear and stone fruit industry, and I'm seconded to Stellenbosch University. I just want to tell you about our cover crop research in apple and pear orchards. It's happening in series, and just as a start, I want to explain to you the sort of basis of which it originated and the sort of philosophy behind it. In terms of ecology, grassland ecology, there is some very interesting work done by David Tillman on diversity, grassland diversity, and productivity, stability and resilience. And it's a long-term trial that was carried out in North Dakota. And at the end of that, he proved very conclusively that the more diverse your plant community is, the higher the primary production, the more sustainable or stable it is through time, and it is more resilient to upsets such as drought. So taking that and applying it to orchard ecology is quite a step, but anyway, we tried. So the idea was to trial cover crops from fairly simple grass only through to diverse cover crops, and it was limited in terms of seed availability. What was actually out there, one would prefer to plant indigenous grasses, for example. They're just not available in bulk. And it took a while to set this trial up. We designed it, I redesigned it a few times. Eventually it was approved. And the trial also involves under tree treatments versus cover crops. Okay, we here, not in the trial site, but at Wes-Kaap in Stellenbosch, that just coincidentally has been planted to a cover crop. I just want to give you an idea of our treatments in series. So we ended up with 4-row blocks of treatment with 4 blocks. Right, so there were 4 treatments, 4 rows. So I had 4 inter-rows or work rows and the associated tree row. So I'll refer to them as work row and tree row. The basic treatment in the work row were 3 grass species. It was a rye and 2 fescues combined with triticale, and just incidentally we put the triticale down at 20 kilos per hectare at that rate. We cultivated our treatments absolutely clean, and I'll get into that later. I don't think it was the right thing to do, but we did it anyway on the understanding we basically false bedded it to get rid of weeds, and then planted. So our first treatment was our stock basic treatment. The 3 grass species, triticale in the work row, and then on the tree row, on the bunkie, we had compost, lumbary compost, and a straw mulch. The second treatment was that same grass mix. Instead of a straw mulch, we had compost and wood chip. The third treatment in the row was the 3 grass species, and then we added a medic and a subterranean clover. And on the tree row, we did not mulch. We put a medic in an attempt to replace straw mulch or a mulch over the compost. And that was done. This is an active debate. Can the industry afford to import large volumes of carbon in the form of straw, for example? It's getting expensive. to put on our soils to benefit them? What about the swartland? Surely that stuff should stay on site. So the importation of large volumes of carbon is problematic, and it's also expensive. So between the farm management and myself, we came up with the idea of putting in a medic or a clover to form to cover in winter, and then it would either be burnt off or dried off in summer to cover compost. Okay, and then the fourth treatment, I was sort of given free range in terms of diversity. So we had the 3 grasses with the triticale in the work row, and we planted an additional 6 species, and they went from things like chicory, yellow mustard we put in there, tillage radish, pea, and more recently we put in phacelia into that mix. And then on the bunkie, on the tree row, we stuck with our medic clover mix to establish. Okay, and that was all compared to a untreated standard practice control adjacent to it. Having said that, there was some issues in terms of, I was unhappy and still am in a way, in terms of, do we know enough about potential cover species? We don't, and I think this is an ongoing element. So I did a series of 3 meter, this is pretty typical, a 3 meter long block in the work row, and I've tested a variety of stuff. I've even put in stuff like lucerne. That's not a good idea, it just takes over. But I've trialed strawberry clover that looks impressive in series. I don't know how it's going to react, for example, in Stellenbosch. Crimson clover looks great. There were a bunch of other cover crops that we put in. Poor man's lucerne, ceradella we've looked at. And I think we're going to end up almost sort of tailor-made mixes of cover crop species to ensure diversity over time. Okay, and the other one is the repeat. Are they annual? Can we treat them as annuals, or are some of them going to be perennial? Coupled to that is cover crops that flower. I'm an entomologist, so I am very interested in flowering cover crops for bees, and more importantly for parasitic insects, wasps in particular. So I've trialed a bunch of those in the work row. In the tree row, we've put in some as a cover. That wasn't very successful, but there's this area. Here's your work row. You've got a tractor tire sort of compression zone here, and you mow up to a point. Then you've got your tree row that comes down. There's an area on that edge in which you can put flowering plants. And what have we trialed? Alyssum. It's a funny little plant that's used as a ornamental ground cover. Is fantastic. It works really well in terms of flowering and persistence. I don't know how attractive it is to our local wasps. There are also things like chamomile has worked particularly well. Dill has worked. Coriander. Those are big umbelliferous plants with big flower heads like this, and small wasps love them. What else loves them, I don't know. And yeah, during this process, thank goodness for the nematologists, and in particular Rinus Knutzer, said, what is the nematode host status of these plants? I said, well, we don't really know. There's some literature, but it's not comprehensive. He has in fact trialed most of the cover crop plants as hosts, whether they're susceptible and support Pratylenchus, lesion nematode, that's really important. And funny enough, one of our really promising cover crop species, buckwheat, that was great. It grows very quickly and it flowers very quickly, is one of a really good host to Pratylenchus. So that went off our list. Yeah, that explains what we did and how we treated it. So I'm working in fairly advanced orchards in terms of they are mulched, and they do have a modicum of flowering species in them, and there is diversity. And before we go further, there are two bits of entomology that are critical to acknowledge. One is biological control of mites in orchards depends on orchard floor diversity. That was established by Ken Pringle 25 years ago, and it hasn't changed. We're having issues now with biological control of mites, and I suspect it is because of the cover crop not being diverse or permanent enough. The other one is mulching gets rid of apple woolly aphid on apples very effectively. So the mulch stroke cover crop debate starts is a living mulch, like a subterranean clover or a medic, going to suppress woolly aphid in the same way as a mulch such as straw would chip. Okay, having described the treatments and how we applied them and separated them, the next issue is how do we monitor and measure this lot? Quite simply, and there will be a visual of it, a 1/2 meter square quadrant is a very useful tool. We measured in 5 fixed monitoring points per treatment on the trials. So we go back to the same place every year or every month. I use a standard monthly monitoring protocol is to take the quadrant, put it down, take a photograph at a set height, funny enough with an iPad that works really well. So I've got an idea of what is growing in that 1/2 meter square. I also take from a standard height, standard position, a long photograph down the row of the work row and the tree row for each of the treatments, and that is critical. My memory is short. One has perceptions about how your cover crop's doing, and it's absolutely phenomenal how it does in fact change. So I take those images, just it might be the easiest way to do it. I just put them into a standard format on PowerPoint with a date, and I write comments underneath it. How that'll pan out over time and experimentally, I don't know how useful it is, but it's incredibly useful for extension and showing people differences, even well, not even, but researchers and farm management. The other one that I really battled with, and some Swedish ecologists put me right on it, was how to measure cover crops. How high is it? You can't get in there with a tape measure and just measure how high it is. And I've got a measuring board, we'll show it to you, that's just marked off in 100 millimeter blocks, 100 millimeter deep, and it goes up to 1.1 meters, and it stands up, and it's a board, and you stick it down, go 10 meters away from it, standard height with a decent camera, and you take a photograph of it, and you can process that image very easily with an image processor to get very, very accurate percentage cover in those blocks. So you know exactly how dense your cover crop is and how high it is. And that works an absolute dream. Okay, so those are the measures on cover crop. The other one I do annually, and I do it at the end or the beginning of November, is with the quadrant, go out in the middle of the row, and I collect all the plant material there for primary production of it. I bag it. I do that 5 times per treatment. I haul it back to the lab and I separate what I have into grasses, into broadleaves, planted broadleaves, that's stuff that we actually planted, and that's clovers and the like, and the other component is obviously weeds, which we haven't discussed, but we will. Anyway, I then dry them in a drying oven and weigh them out. And just coincidentally, my diverse cover crop did give the highest primary production, and we went up to just over a kilo dry mass per hectare, per square meter. Sorry, that translates to 10 tons per dry mass per hectare, which is basically what we need. Other measurements that we're doing, we take soil samples at those 5 sites per treatment, we pool them, and we do a Solvita soil test on them. If you're interested, have a look at Solvita. It's run out of Ward Laboratories in the States, and it's an incredibly comprehensive soil test, and it's got a lot of biological parameters are also measured in there. I find it extremely useful. There are other diversity. I'm interested in diversity and the biology in the soils and the effects of the treatments. So we're also looking at the diversity of nematodes through NemLab. They've got 7 functional groups of nematodes. They don't all eat your plants. Some are fungal eaters, some are detritus feeders, some eat insects, some eat nematodes for that matter, and some are just free living. They classify them and they give you a diversity index that's incredibly useful. I'll get to it later. We also look at the numbers of damaging or pest nematodes, such as Pratylenchus. So these samples, I should note, are taken under tree and in the center of the row at the same point. So we have a comparison of the effect of the tree row treatments versus work row treatments. Recently, last season, out of that sample, that self-same pooled sample, we monitored fungal diversity and had a look again at the diversity of the different groups, and that's run through Sporotech. Again, difficult to interpret in the first year. It'll be very interesting to see if it does shift in the second year, but already you can see groupings across the certainly the tree row treatments. Remember, they all get compost, so they should be very, very similar. They're not that similar, funny enough. Is the effect of the plant diversity in the row on the fungal diversity? We don't know yet. I'll be able to confirm that after November. What else are we measuring? Obviously, I'm in the fruit business. I'm not in the cover crop business, so we'll be looking at tree growth. We'll be looking at production. Do I expect to see a difference between the treatments? I don't think so. There's more soil work to be done in terms of aspects such as soil moisture. We haven't addressed that yet. And aspects such as water ingress into cover crop planted soils. There are also other aspects such as litter breakdown that I'm very interested in. Litter is one of those ecological parameters that a lot of stuff lives in, and predaceous mites, mites that eat the mites that get on your tree, live in there over winter. What do they eat? We're not sure. We've got a litter trap. It's quite a small thing with a coarse mesh on top and a fine mesh underneath, and we put a measured amount of leaf litter in there, and we bury it at soil surface. That has the advantage, one can get two parameters off it very quickly. One is litter breakdown. How quickly do the leaves actually break down? It's important, I'll come back to that. And the other one is what is in there. And there are a whole lot of very small little insects and mites that get into that litter, and we can measure them. And there's industry-funded research looking at that right at the moment, right down to how carbon is going to be transported from litter layer into insects and who's eating who. It becomes quite complex fairly quickly. In summary, yeah, the results they're preliminary, they're very complex. It's one thing about cover crops and orchard floor ecology. It is very, very complex. I think we're making headway. Every year adds on to it, and we're producing, for the first time, we've got 2 students actively involved that should help substantially. The aspects, as soon as one starts research, one's sort of locked in for a while to the methods, but during that process you sort of develop thoughts and the farm management that's involved, and there are a bunch of groups out there who are all looking at cover crops, and there are lots of variations on the theme coming out, people using different species. There's one aspect that really interests me, and I think it would be a very worthwhile investment to look at it, and that is the use of indigenous plant species in orchards and vineyards for that matter, and outside in grazing for that matter as well. Sharing that information, there are a number of people who are very interested. I've planted some, they haven't been very successful. The other one is, what is the effect on our production practice, or what do we need to change in terms of our production practices to allow cover crops and the benefits associated with them to actually be realized? And there I'm talking improving soil health to the extent that you start cutting nitrogen bills, your water ingress, water retention improves, and you can start manipulating pest populations downwards by the use of cover crops and mulches.